goals for basal ganglia. Yeah, so I have come up with a brief agenda for today's session. There might be ups and downs within the agenda based on how the session will take over. But yeah, this is the overall agenda for today's session. The first thing we will be starting with is understanding what is the decision-making deficits that you actually see in Parkinson's disease, or basically what is Parkinson's disease and what are the kinds of cognitive disorders you will see with them. And next, we'll actually go through the entire architecture of basal ganglia because that is what we are going to use it for simulations today. So we'll be understanding the overall pathway within the basal ganglia. Here, BG represents basal ganglia. Then we'll also look at one perspective of basal ganglia, which is how is reinforcement learning taking place within basal ganglia, or basically reward-based learning being occurring within basal ganglia. Then we'll actually take up one task, which is a gambling task that is called as Iowa gambling task, and do some kind of simulations so that to get a hands-on on how does the simulations work and how does the performance vary as we keep varying certain parameters. After that, there is one key component within the basal ganglia, which is STN-GPE system. We'll actually try to model the entire STN-GPE system as a 2D grid of network of spiking neurons and look at its behavior. Then we'll also study how do we model the deep brain stimulation aspects of STN and how do we actually do or look at the simulations. At the end, we will go through the simulations of STN-GPE system. Once we do the code simulations, you can actually look at how does the normal behavior of STN-GPE system looks like and how does the PD behavior of it, that is Parkinsonian behavior of it looks like, and how does the Parkinsonian and DBS behavior looks like. I mean, PD with DBS behavior look like. Then we'll actually integrate the entire STN-GPE system into the overall basal ganglia architecture and again simulate IGT and see how does the performance look like. Like this is the kind of overall agenda that we will be doing today. I don't know if we will be able to finish the 8th one completely, but again, we'll try to finish it up entirely by the end of the session today. So in a way, these are the goals. Maybe I'll talk a little bit over the session and then go through the goals because these might sound very random at this moment. But I'll go through the goal of the session towards the end also to pick up how many goals are we able to achieve with it. So starting with the decision making in Parkinson's, so what we have done is the entire session, we have segregated it into some parts of theory so that you can understand the basics of all the modeling and also the biology behind it. Then the code simulation, following by theory and code simulation. That is how we have organized the session. So we'll have certain theory components also within the session. So just bear with that. Yeah, so introducing decision making in Parkinson's disease. But first, let's understand what is Parkinson's disease. Parkinson's is basically a neurodegenerative disorder. When we say neurodegenerative, it implies certain cells are basically dying. And those cells are nothing but dopamine cells, which are present within substantia nigra pars compacta. Now, the name sounds very this way, but it's basically a small neuron cluster, which is present within one subcortical structure, which is basal ganglia. So due to the cell death, most of the symptoms of Parkinson's actually keep showing up. Now, since the cell death occurs within basal ganglia, let's also try and understand what are the structures that are present within basal ganglia. When I say structures, now entire basal ganglia is a full structure. Within that, there are small clusters of neurons, which we call them as some other structures, which we'll look at it. Now, if we look at entire basal ganglia as an input-output system, there is an input port to it or an input structure to it through which the information passes through, that we call it as striatum. And there is an output structure to it, which we call it as GPI or globus pallidus interna. Apart from these input and output structures, there is of course the substantia nigra pars compacta, which is basically dopamine producing neurons. And also there are two other key structures, which are subthalamic nucleus and globus pallidus externa. So these are the 5 very important structures which you keep coming back to all the time when you're actually having a discussion. Now, Parkinson's is majorly classified as a motor disease. That is, they'll show issues with movements. They can't move their arms very easily. They show tremor, which is basically shivering kind of a behavior of hand. They also show bradykinesia, which is like slowness in movements. They have changes to postures. They can't stand still. They have a stooped posture. Their stiffness is also pretty high, and their facial expressions are also low. And generally, when you go to clinics, Parkinson's is treated or characterized by motor symptoms, and people only try to treat these motor symptoms. But apart from motor symptoms, there are a lot more other cognitive symptoms which actually come way more before motor symptoms actually show up. I mean, the entire PD motor symptoms show up only after 70% of the cells are dead. But cognitive symptoms are, at least few literature suggests that cognitive symptoms are shown before the motor symptoms even arise. So it becomes a far more easier thing if you are able to identify the cognitive changes before. Now, when I say cognitive changes, there are many aspects to it. It can be memory, it can be attention, it can be decision making. But today's session will be entirely focusing on decision making alone. Just for simplicity, because we can't model all the cognitive aspects in one session. So we'll be focusing on decision making. Now, decision making is again a very core function we need every day. That is, decision making can be very simple, as from taking different food choices, what kind of food I need to pick for this time, to a more complicated decisions like which stock do I invest in. So it becomes very integral function, and that gets disrupted within Parkinson's patients. Now, when I say decision making is disrupted, what are the actual functions that are disrupted? First thing is they show a lot of impulsive behavior. That is, they have a lot of impulsive shopping behaviors, or they have a lot of gambling kind of behavior, and they end up losing a lot of money. They also show a lot of risk taking behavior. That is, if you give them a choice of picking from a high risk stock versus a low risk stock, they end up always picking up the high risk ones. And their understanding of reward, punishment, and also the probability is very, very poor, because of which they can't make decisions very easily. And also, they can't do delayed discounting. That is, they can't estimate the futuristic reward they are going to get and make decisions right now. And their flexibility and set shifting is also poor. By set shifting, what I mean is I give them one paradigm of task to perform and suddenly switch the task paradigm. They can't adapt very easily. So these are a lot more broader perspectives of PD where they actually showed decision making deficits. Today, we will be only discussing on gambling. We can actually model all of them using this very same network. But for simplicity, we'll be taking one classic task, that is Iowa gambling task, in today's class and today's entire session, and see how it goes. Yes. So, as I mentioned, under literature, people have studied a lot of Iowa gambling task. It basically tests your decision making under risk and ambiguity. The task goes like this. It basically has 4 decks of cards. Few are highly risky, few are low risky, and PD patients show a high risk picking tendency. Like when you give them a choice between these high risk versus low risk cards, they actually see or seem to pick a lot of high risk cards. Now, when deep brain stimulation is given, they don't really show much of a difference. They rather showed either worsened or no improvement. I'm assuming people understand what is deep brain stimulation. If not, maybe let me just go through a little bit. So, what people do is at the later phases of Parkinson's disease, when you can't treat them with medication anymore, they perform a small surgical surgery on the patient, implant electrodes in the subthalamic nucleus. They go inside the brain, they insert electrodes within the subthalamic nucleus and keep stimulating it with some high frequency stimulation. That actually is shown to improve motor symptoms, so they keep doing it. I hope that sort of clarifies what is deep brain stimulation. It's basically a therapeutic method where they surgically implant electrodes and keep stimulating STM, especially subthalamic nucleus. And people have tested it on motor symptoms and it works very, very well. But if you actually try and look at it within decision making in itself, it doesn't show any improvement. That is, when I do deep brain stimulation, which is good for motor symptoms, doesn't really improve for cognitive symptoms. So that is what we are trying to address today, why it should or why it shouldn't be improving today. Or why does the current protocol doesn't really work out? Maybe that is what is the question you will be trying to chase till the end of the session. Yes. So to understand or to even begin modeling such a decision-making behavior, first let's understand how do we make decisions to do something. That is, assume I have a couple of stocks and I have to decide where to invest. What are your steps that you would be performing to decide where to invest? Can I see the chat? Yeah. You guys can answer on the chat boxes itself. How do you decide? Where do you invest? Or you can unmute and answer as well. Given a choice of investing into a couple of stocks, how do you decide? Yeah,
Probably,
yeah.
probably we'll check the market and like see which stock is going up or down, like the current trend.
Mm-hmm,
And other than that, maybe there would be some stocks that are relatively high performing all the time. For example, Amazon, Nvidia stock
mm-hmm,
is going up right now,
mm-hmm.
something like that.
Yeah. So the first thing we might tend to do is look at its past history, right? Some kind of value or a metric we will look at to understand if it is actually a good performance or a bad performing stock, right? Say for now, let's keep it very simple. Maybe we will actually try to look at the price history of it or returns, how much returns it has offered in over a long time, and make a decision. Now, do we always invest in the ones which are highly performing, or do we also try out other ones in general? Maybe not every time, but sometimes we also do tend to invest in some other stocks thinking that they might improve, right? Does that make sense? Yes.
Yes, ma'am.
So this is what is a behavior in general, which we tend to call it as explore and exploit in very classic reinforcement learning literature. So basically, when I say that I invest in the one which has been like giving consistently good returns in the past, then I know that I'm exploiting the known knowledge of how much returns I have been having, which I call it as exploitation. Apart from those, if I try out some other new ones, then I call it as exploration. That is how any decision making takes place. And the balance of this exploration and exploitation is very, very important. Don't you agree that exploration is also very important in general to make any decision? If I don't actually explore, how will I even know which is the best one that is available? Sometimes we might actually have to explore to reach the optimal most investment. So both of them are very, very important. So if you want to think of a very another very day-to-day example, we can think of it as restaurant picking. I can always go to my favorite cafe, which I know that serves best food, which is exploitation. But I can also at the same time try out new restaurants that are available in the town, which is actually exploration. Now, why is it important? If I don't try out the newer restaurants, how will I know whether the restaurant I always pick is the best one or not? So, always any decision making involves this exploration and exploitation as two very key compounds. And the other thing is value. Value is basically average expected reward or return. Basically, when you are even looking at your stock investment, you actually look at some kind of returns, right? Over time, this is the average return I have got. Based on that, only I can make a decision. Similarly, value is nothing but average returns that are being available, and it is very, very important. And these are very classic terms that are used across reinforcement learning literature, but I am keeping it very standard for this specific session. There are more formal definitions of it, which you can go through when you're doing any RL literature. So, yes, with that understanding of decision making, now we actually know that any decision making requires these two things: exploration, exploitation, and value learning. With this understanding of basics, maybe let's start looking at the biology of basal ganglia architecture. How does the basal ganglia architecture looks like? Now, this is the overall BG architecture, the way it actually looks like. This is how they put up in general papers, but you can actually look at it as a simple input output kind of a network. You can think of that there is some input, which is a sensory input or any kind of state input, etc., that goes into the cortex that enters basal ganglia through its input part. The input structure is also called as striatum. Now, striatum has D1 and D2 separate neuron clusters. Okay, so the input goes through the cortex, then enters the input structure of your basal ganglia, which is D1 and D2 striatum. From here, using two different parallel pathways, it reaches the output structure of the basal ganglia, which is basically GPI. From there, it goes to thalamus. From there, it leaves the structure. So there's the input structure, output structure. How is the information processed between these input and output structures? There are two parallel pathways that go through. One, we call it as a direct pathway, which connects input directly to output. And the other, we call it as an indirect pathway, where the input and output are connected via this STN and GPE system. And STN and GPE also, if you look at it, they form an inhibitory excitatory loop. That is, GPE is inhibitory and STN is excitatory. You can actually see that red arrows are inhibitory and green ones are excitatory. And SNC, substantia nigra pars compacta, which is basically a dopaminergic dopamine producing cells, actually project onto this D1 and D2 striatum. They also project to STN and GP. We'll look at it later. But yeah, this is the overall architecture. Now, if I just consider the direct pathway where the input goes from cortex to striatum directly to GPI to thalamus. Remember, the red arrows are actually inhibitory. What do you think will be the overall effect of direct pathway?
Inhibitory?
It should be in a way excitatory, right? Because there are two inhibitory conditions. That is, at the level of thalamus, there is one inhibition followed by another inhibition. Can I say both of them multiplied, it will give excitatory effect?
Yeah.
Yeah. So the overall effect of direct pathway is basically excitatory. That is the reason we also call it as go pathway. It promotes a movement. If I want to reach some target, it promotes that specific movement. Similarly, if you actually look at the indirect pathway, this is how the indirect pathway looks like. It goes from D2, which is inhibitory, then GPE, which is inhibitory, then to STN excitatory, and GPI, which is inhibitory. What do you think will be the behavior of indirect pathway then? Overall effect.
Neutral, maybe.
Inhibitory.
It's inhibitory. So the way we can understand whether it's inhibitory or excitatory is you go towards that end. This is mathematically how you can look at it. You look at that forward pathway. There's 1 minus 1 minus. You just take a product of it. Negative times negative is basically positive. So the overall effect is positive in direct. Indirect, if I look at the forward pathway, I have negative here, then a negative here. So negative into negative is positive. Then I have a positive here, positive times positive, positive, positive times a negative, which is negative. So it becomes inhibitory. That's how you can look at it at the circuit level if you want to do mathematically. So the overall effect of indirect pathway is actually inhibitory, which is it will stop movements. If I don't want to make an unwanted movement, it will stop it from making a movement. That is the reason we also call it as go pathway as well as no go pathway. Any issues still here? You guys can stop me right here. Otherwise, I'll go ahead from this point. What is the input? Visual information or the action plan? You can take it as visual information, or if you're playing a game, you can understand it as a state input. I don't know if you understand what is state. The current state can be the type of stocks. Say I have N stocks. What are the stocks available? That can be your input. Or if it's a motor task, I can say the visual information of my target is my input. So input would be task dependent. Does that answer your question? Yeah. Okay, so basically this is how the overall network architecture looks like. Now what happens is dopamine, that is SNC, which projects onto this D1 as well as D2, has a kind of differential effect. That is, whenever there is a higher dopamine present, there will be a higher direct pathway output. Whenever there is a lower dopamine present, there will be a higher indirect pathway effect. Now, in PD, what happens? We'll see that dopamine cells start dying. That is, there is lower dopamine. So what do we expect? Overactivation of direct pathway or indirect pathway?
Indirect.
Indirect pathway, right? So that creates a lot of circuit imbalance. Generally, under normal condition, indirect pathway gets activated whenever basically you have to stop any random movement. But now under PD condition, due to dopamine loss, what happens is the indirect pathway gets overactivated, creating this circuit imbalance. because of which a lot of symptoms come up. So technically in indirect pathway, one very, very important structure comes up, that is STNGP. And these dynamics are very well studied across literature. So let's also have a look at brief understanding of what happens to the biological dynamics of STN and GPE. So let me go through that. Yeah, so if you look at the evolution of understanding of STN-GPE system, as I already mentioned, the indirect pathway gets overactivated. So initially, people also started thinking that STN also starts behaving overactive. They also tested it experimentally by surgically putting electrodes and testing. When I mean overactivity, they observed that there is an increased activity at STN. Now, due to which, when there is a higher indirect pathway activation or a higher STN activation, you remember it leads to a lot of no-go movement, causing a lot of hypokinetic or very reduced movements. Now, they usually used to think that DBS used to reduce, or the deep brain stimulation which is given to STN here, used to reduce this overall activity, and thereby it releases the movements. That's how DBS used to work. Then they actually, instead of saying randomly that it just increased the activity, they actually looked at what kind of activity happens at STN. And they observed that there are synchronized abnormal bursts that happen at STN, specifically in alpha and beta frequency, which is 10 to 35 hertz. Do you guys understand by the term what is bursting? Bursting is basically some kind of chattering behavior. That is, all neurons fire together and stop, fire together, stop, fire together, stop. I'm sure you guys would have seen it in day one tutorials or even in the sessions. One kind of neuron firing is where it fires, stops, fires, stops. Such kind of bursting activity around 10 to 35 hertz is actually seen. That to synchronize. When I say synchronized, all neurons fire together or stay silent together. Then they figured out that DBS goal was to actually reduce this beta synchrony, which is why it was able to improve the symptoms or the motor symptoms. But again, when I say it improves motor symptoms, its effects on decision making is not at all very great. It doesn't improve, or it rather worsens. Or in 90% of the times, it doesn't really improve any kind of decision making. So to understand why it should not improve, or at least to understand why does the current DBS protocol doesn't improve any decision-making performance, we have to understand the reinforcement learning perspective of basal ganglia first. Then you will do some kind of simulations where you'll be able to figure out why does actually DBS doesn't improve the performance. Now, you remember that there are two very key important elements of decision making we discussed, right? One is exploitation and the other is exploration, and the other is your value learning. All of these three things are very important. Now, they should be happening somewhere within the basal ganglia architecture, right? We have seen that go, no go pathway. All these elements should have been done somewhere within the basal ganglia, right? Does that make some sense? Because basal ganglia is known for doing reward-based learning and decisions. So it should be done somewhere. So if you actually look at the entire architecture, you can slightly rephrase the architecture of go and no go to look at it in terms of explore and exploitation. That is, if you want to understand exploitation, the direct pathway is very good or promotes exploitation. Now, for even doing exploitation, I need to estimate value. If I know the recent returns, then only I can estimate the value, right? That value gets computed at D1 striatum. In general, that is where the value gets computed, and the direct pathway promotes exploitation based on that value. Saying, okay, fine, out of these three actions I have, action A has higher value, so better go for that action. What about exploration? Exploration actually comes up from the random or the complex dynamics of STN-GPE system. Now, why I say complex is, do you think exploration is random or very strategic in general? In general, we tend to explore randomly, right? If I have 4 new choices, I will randomly pick all the 4 choices and see which one works best. So randomness is very important for exploration, and that is what comes out of these STN-GPE dynamics. You can think of it more like that. So basically, this is what I mean. The regular go, no-go view might overlook the exploration part, But the exploration actually is a result of the complex dynamics of STN-GPE system. That is, whenever I have higher complexity, I'll be having higher exploration. Whenever there is a lower complexity, I'll be having lower exploration. Now, under PD, I said that there is a lot of synchrony. That is, its complexity is lost. Can I say exploration is lost because of that? Yes, right. When I say that the entire STN and GPE system has become extremely synchronized, all of them are firing together. There's no randomness in the system, due to which actually the complexity gets lost. In a way, you can think of it like exploration is actually lost within your STN GPE system. And DBS is actually supposed to restore that complexity in a way. Does it do it? We can have a look at it when we actually do the simulations in itself. Any doubts still here? I'll pause for a minute here and take up any queries.
Ma'am,
Uh,
how did you?
Yeah.
yeah, so can you explain it again that how high synchrony leads to disrupting exploration? And when you say high synchrony, like you're saying that all are firing and synchronized, so how does that thing happen in Parkinson disease?
Yeah, so when I say all of them are firing together, if you actually look at the rate output of all these neurons, all of them look the same. Or rather, I would suggest if you take 4 neuron clusters within STN, and when you take them under synchronized condition and look at the variance that it creates across these 4 clusters, it's very, very low. When the variance is low, can I say the randomness within the network itself is low? You'll see these numbers produced when you're actually simulating spiking network, but when the variance across these 4 or 5 clusters within STN is low, can I say that the randomness within the network in itself is low? Does it clarify
Yeah.
something? Yeah. Now, when that randomness is low, it can't really explore. See, exploration is pretty much random. When I have 4 outputs and I have to explore across all 4 of them, I will randomly sample from these 4 choices, right? Say some person might pick the second choice, some person might pick the first choice randomly. And I'll try out all the options, then only come with the exploitation. That doesn't really happen under PD.
Yes, now it makes sense. Okay.
Yeah. Any more
Yes, thank you.
queries here? Thank you. Yeah.
How did you say that exploitation is like a direct pathway? Is it like less
Um,
complexity, so you consider like a direct pathway requires, I mean, direct pathway equates to exploitation, and since more complexity is required for exploration because of randomness, considering the randomness of decision making, you consider like exploration as an indirect pathway.
yes, that is one. And the other reason is to exploit, don't you agree? I need to estimate value first.
Yes.
That value gets computed in direct pathway. Basically, that D1 part of striatum, people have actually recorded stuff saying that it represents a value function. So what it says is out of 4 actions that are possible, it will keep computing value for A, B, C, D. Say A has a value of 10, B choice has a value of 5, C has a value of 2, D has a value of 15. So direct pathway will keep saying, okay, fine, D has a higher value, go for it. The indirect pathway will be like, even though it has a higher value, why don't you try out other choices as well?
Okay, ma'am. Now I understand. Yes, thank you.
Yeah. Any more queries before we go ahead with it? Okay. Let me go ahead a little bit. So now to understand the training part of it. So how are we going to train the entire vessel ganglia network for any of these reward-based learning tasks? So this is how we do. All the structures, all the layers here are basically taken as feed-forward network layers. You guys have gone through deep networks, right? So you can think of all these as simple deep network layers with sigmoid activation. Apart from STNGP, STNGP will come to it slowly. How can we model STNGP and stuff? But for now, all the other layers are basically your deep network layers with sigmoid activation. And the value function is basically taken as this output of your D1 pathway. This output that connects to D1 to GPA is what is we take it as value. Here we call the value as Q value because we are taking value for each of every action. So we call it as Q value. Now, when I have a value, there should also be learning, right? See, I will always have a predicted value, which I have to always match it to the true value. Agreed or not? Yes, right? I'll always have a prediction that I have made. And I also look at the current true value that is possible and have to keep taking the error of it and keep training the network. That is what we actually do. Here, R is the true reward, value is the estimated reward. We take the difference between it, which we call it as temporal difference error. And we use that basically to train the entire network. You know that we need a loss function to train any deep network, right? So this is the error we take as training error, with which we are actually training it. At the end of the day, what happens is your true value will get very much equal, true reward or true return will get very, very close to your estimated value, which is what we actually want. And that is what this keeps doing. Is it clear till here? Now, this TD error is very, very important in literature because people have done studies, especially Schlutz's experiments suggested that this temporal difference error is what represents dopamine in brain. Basically, whenever you have a value estimate and whenever there is a true reward that is given to you, and whenever there is a difference between them, the dopamine cells start firing. And that is what is basically represented as dopamine. So TD error is a replica for dopamine. And whenever we do modeling studies and to replicate PD condition, we'll try to limit this dopamine itself. So if you actually want to look at what happens under Parkinson's condition, so generally the network is trained using this, but how do I replicate PD condition within this training network? Basically, under PD condition, the dopamine gets depleted. So basically, we have to limit our delta as well because delta is dopamine. So we will limit our dopamine to some upper cap value. It can't fire beyond a limit because some part of the cells might have died. Before it might have fired for 100%. Now it will only fire for 50%. Apart from that, the dopamine also has an effect of ST and GPE dynamics. It ends up showing synchronized dynamics at ST and GPE. Basically, when I say synchronized ST and GPE dynamics, I'm saying that there is no complexity at ST and GPE dynamics. So if you want to look at entire Parkinson's behavior, what is exactly happening with it, there are two impairments that occurs at PD. One is value impairment and the other is exploration. Exploration, I have already explained why it happens, because the complexity at STM-GPE system is lost, the exploration is lost. Since the TD error in itself is reduced, the value impairment occurs. Does that answer most of your queries? Why I say value impairment is if my TD error or the error which I use to backpropagate the entire network itself is wrong, can I say my values learned might not be correct in a way? Yes, right. If my error itself is high, like error itself is wrong because I have limited my error. Initially, it was supposed to go to some 100%. Now it only goes to some extent. Your value estimations will be not really correct. So they also show value impairments. But for today's session, we will be restraining ourselves with exploration deficits. We won't be discussing in detail with value impairment, though the code can still simulate the value impairments. We are only focusing right now on exploration deficits. Is that okay?
Hello, ma'am.
Yes.
You mentioned complexity, right? Can you
Yeah.
give a context or what it actually means? I'm not so sure what it means.
Yeah. So for now, imagine STM-GPE system. Don't think of it as an entire neural network. Think of it as a simple noise generator. Maybe our first
Okay.
experiment will be like that. So think of it as a simple noise generator. Okay.
Okay.
Think of the complexity as the variance across the noise. Higher variance,
Okay.
higher complexity. Does that make more sense?
Okay. Okay. Fine.
Yeah.
Okay.
If you have done some kind of reinforcement learning, you would have seen that for exploration, you actually do some kind of epsilon greedy or something, right? Where you actually have with what range of noise can we change the exploration karke. It's more like that.
Okay.
Yeah. Any more queries? Okay, so for now, what we will be doing is for the first part of the session, to not confuse you guys a lot more, we won't directly go on to the STN-GPE network itself. We'll just have STN-GPE network as a simple noise generator because exploration should come up from noise itself. We'll think of it as a simple noise generator with certain variance that is present. Think of it like a simple np.random.random with some variance present and simulate it on a decision-making task. At different variance levels, we will see what happens to the performance. That should be the first part of the session today. That will give you an understanding of how variance that is present at STN-GPE will give you better results. Okay. So to even model the network, we need a task, right? So for that task, one task we have chosen for today was Iowa gambling task. As I mentioned, it's a very, very classic task that is tested across cognitive community. So it has this. It basically has these 4 decks of cards, which we call it as A, B, C, D. And there are 2 decks of them which are actually bad. There are 2 decks which are actually good. The goal of the person is to keep playing and identify which are the good decks. We won't tell them what are the good decks and what are the bad decks. We will only tell them, okay, fine, you play and try to maximize your reward. Okay, so how are they bad decks and how are they good decks is like this. Say for my card A and card B, I give a reward of 100 and 100, but I also give card A with a loss of minus 250 with 0.5 probability and a card B of loss minus 1250 with a probability of 0.1. Similarly, card C and D gives a smaller reward that is 50 and gives loss of minus 50 and minus 250 with probability of 0.5 and 0.1. If you actually keep picking card A and B, even though the reward is high, the loss is also very, very high. So you'll end up losing a lot of money in that. Similarly, C and D, though the rewards are lower, you won't really lose money because your losses are also lower. So you'll end up having a positive return, which you won't understand when you're actually playing it. You will have to keep estimating your number on your brain and then only play it. Okay. So this is kind of your high risk and low risk stocks in a way. You can think A and B are high risk cards and B, C and D are low risk, more gaining cards. So to evaluate, what we do is we compute something called as an IGT score, which we take it as number of advantageous cards minus number of disadvantageous cards. I make a person play it for 100 times and keep looking at this IGT score for every 20 times, which we call it as basically wins. So this IGT score is how many times did I pick my advantageous cards versus how many times did I pick my disadvantageous cards. Now, if my score is actually positive, implies the person might have learned, okay, fine, these are more advantageous over time. If my scores of IGT are negative, it implies that my picks are more disadvantageous and they weren't able to distinguish between these cards very well. And generally, even experimentally, we'll end up seeing some kind of a positive scores for normal and negative scores for PD in general, or normal will have a higher score in comparison with PD. Is that okay?
Yes, ma'am.
Cool. So we will go through a coding simulation for a while now. The thing is, as I mentioned already, we will be replacing the entire STN-GPE instead of modeling it as a full network. We'll be simply modeling it as a simple random noise. Can I say in the random noise, when there is low complexity or low variance, it's kind of PD condition. When there is higher variance, can I call it as a normal condition? As an overview, because we know that under PD, the complexity is actually lost. So we'll be doing those experiments as start now. Okay, so the task I want you guys to perform at least today would be the STN-GPE system. There is a noise variance parameter in the code. You have to keep changing those noise variance parameters and see how does the IGT perform vary. That is, I try from lower variance to a higher variance and see how the performance varies. Is the task clear? I'll explain more stuff. How do you access the code and all kinds of stuff? Okay, then we will move to the code first. Let me just... Okay, yeah. Can you see my screen? We have shared the Colab notebook with you guys. Are you able to access the Colab notebooks? If you guys are facing any issues, let us know right now. We have sent the Colab notebooks link in the chat box. I'm sharing it again so that you guys can have a look at it. Okay. So basically, in the interest of time, I am not going to go through the actual code itself. You can open the Gradio version of the code. It opens a simulator. If you are interested in doing a full code simulation yourself, then you can open this IGT noise simulation. Or if you are better with the interface, you can open the IGT Gradio. It opens a simulator, so it's easy for you guys to play around with the parameters. I am right now using the Gradio version. You can also use the noise simulation. In that, you will have variants where you can change it. It's up to you guys. But like the Gradio file is easier to use, you can use that. Let me simulate it myself first. If you are able to open that IGT Gradio file, just simulate it from top to bottom. Once you're done simulating, let me know. Then I will walk you through what is it about and how is the code being simulated. Okay. So basically, if you look at the code, the first block of the code is to clone onto a repository. We have shared a code repository link, which we have made for the project itself. It has this folder. The repository is named from STN spikes to choices. That is basically how does the STN spikes influence your decision making in itself. If you open it, it has like these code modules. The first one is Bessel ganglia. This is the block where actually it implements your Bessel ganglia network. If you open it and see it's a Bessel ganglia network is a simple neural network. And you can also see under the forward loop, you can see that STN can be a simple noise generator. And similarly, environment folder has the IGT environment in itself. Is that okay? Okay, so basically it is cloning onto this repository to basically implement your system. And once you simulate the Gradio file, you can open this localhost link. Is everyone able to access the link? Okay, cool. So as soon as you open, this is how the interface looks like. For now, don't click on this. Use STNGP based simulation instead of noise. If you don't click on it, it will be simulating with noise. If you click on it, it will integrate it with the spiking network, which will do it to the end of the session. Right now, we are only going to conduct the noise experiments. So if you look at the parameters it asks for, you're only going to focus on variance of STN parameter, where, as I already mentioned, we think of it as a random noise generator, which is a random normal noise. We will keep increasing the variance of it and see where the performance improves. That is the task. Is the task clear to everyone? Say if I run it at 0.01, I am keeping the variance of STN at 0.01. I click on run IGT. I'm just waiting for the simulation to be done.
Ma'am, if I'm not wrong,
Yeah,
higher variance means more exploration, and when
yeah.
it's lower variance means they have PD or they don't explore that much. Right?
In general, yes. For now, don't even think of it in those lines. Just increase the variance slowly and see you will see the performance changing. When
Oh,
we do spiking, you can actually correlate these variances with the spiking network variance and actually correlate what is PD and what is normal. For now, at
oh,
least if you can do that much, that's good. Hmm.
okay.
Yeah. So if you actually run for a specific variance, we'll be having different IGT scores plotted up right this. You might not be getting the exact score here. Like if I'm getting -5, might not be possible. If you run it again, you might be getting -5. You might be getting -7 also. But overall, you will see either positive scores or a negative score. If you actually do a statistical analysis, they won't make a difference. Why do you think if I run it again, there is a difference in the value produced? Anyone? That is because of the STN's noise, one. And secondly, it's also more natural behavior, right? If I play the game, the kind of behavior I will show is different from the kind of behavior you will show. I might be ending up getting one kind of a score, you might be getting another kind of a score. So it becomes more realistic to do it like that. Okay. So you will see how the cards are being picked and what is the IGT score. Generally, we take a note of the last win IGT score. That is, last 20 trials, I would have computed that advantages minus disadvantages and picked the scores. And these are the metrics it will print, so you don't have to bother about it much. But can I say that the final score is around -5? Do you think they picked a lot of advantages cards or disadvantages cards?
Disadvantages cards.
Disadvantages.
Yes, they end up picking more disadvantages cards. So what we want to do right now is, if you open this folder, can you see this folder? There is a presentation given to you guys. Can you open it? It opens up like this. Yeah, it opens up like this and it gives you what is the task to be performed. So basically assume that the entire STN GPE system is a noise generator. We keep increasing the variance. You can keep increasing it from some 0.05 to some 0.25. You can increase it beyond that also, but still, let's just stick to this value. And you will try to see how does the IGT performance change. Okay. So that is basically the task. Run it for these variances and note down the IGT performance and understand what is the trend of it with respect to variance. When I say trend, maybe just note down your last bin score. This should be enough. We can do it as a group activity if one person is willing to share your screen and do the simulations. We'll help you guys with the simulation, but you can share the screen. Just make a copy of this presentation for yourself and you can run it and we can save the values in the screen. Is that okay? Is anyone ready to share the screen? Instead of me sharing the screen and doing the simulations for you guys, if one person can share the screen, it's just changing the variance, running it, and noting down the values. We'll analyze the trend together later.
Ma'am,
Yeah.
So the IGT Gradio is not completed for me, ma'am. It's still executing the code. It's taking so
Okay.
long. Should I have to put any accelerator or something?
No, not needed. I think it's also your internet speed. Since you're running it from Colab, it's your internet speed.
Okay.
Or maybe just reload it once and try running it again.
Okay.
Yeah. Yeah. Is anyone ready to share the screen? Or maybe I'll
I
pick
think
someone.
it's I
Yes,
think it
Avi.
keeps running. I think it keeps running, but you can still use the radio, I think.
Oh,
Oh,
here,
is it?
yeah, you can just click on this localhost link and open it. It keeps running because it has opened the page.
Okay.
You open the page and run it. There only it will show the results. Here you don't have to do it.
Okay. Okay.
Yeah. Let me stop my sharing. Yeah. Is there any volunteer who can share the screen? Or maybe I'll pick some names. We'll guide you through how to do the simulations.
Yeah, maybe I'll try.
Yeah, sure.
What am I supposed to do first? Like, should I
Uh,
copy that?
So just make a copy of the code notebook in your drive. Maybe you can share the screen. I can guide from there. That's okay. Can you share your screen, Abdul? Maybe then we can guide you through how to go ahead with the simulation itself.
Yeah, just a moment, man.
Yeah,
I'm
sure.
just creating folders and stuff.
Yeah, sure, no problem.
Yeah, I hope you can see this.
Yes. Can you open something? Right now it shows, I think. Yeah, we can see. Yeah, good. We can see your screen.
Okay.
Yeah. So the thing, you have opened your Gradio code file, right? Once you ran it, you opened the interface.
Just a moment.
Yeah, sure.
Okay, yeah. Video code.
Yeah. So can you do one thing? Make a copy of that presentation in your drive first.
Mhm.
Go to that presentation once so that we can enter stuff and it would be visible to everyone here.
Yeah.
Yeah. Just
Okay.
go to file. Uh, make a copy. You can directly make a copy.
Mm.
Yeah. Yeah. Can you go to slide 5?
Mm.
Yeah. What we will do is we will run it
Okay.
for different variances like 0.01, 0.05 and stuff. We'll enter the fifth bin IGT score in this slide. Is that okay? Just for our reference to see how the values look like.
Hmm.
Yeah. Can you
Okay.
go to your Gradio page now? Yeah. Maybe we'll start with 0.01. We'll take like 5 values and run it. Yeah.
Yes,
Okay.
very bad.
So our score is somewhere like minus 12.5 or minus 13.22. You can
13.
look at the IGT metric there. It'll give that last bin score also. Minus 13.
Yeah.
Yeah. We can note it down in your slide also. For variance of 0.01. Yeah. Can you run it maybe for what? We'll run it for 0.05 maybe. Yeah. It's minus
Mine.
7.9. Uh, can you also run it at 0.07 maybe? That might be useful later. So just
Okay.
yeah. It's a minus 4.9. Okay. Any more numbers you want to try? 0.1, 0.15, anything you want to put up? We'll go with 0.1. If anyone has any other numbers you guys want us to try, we can. We're just gradually increasing things and trying it out. It's 2.3. Yeah. Then we'll do the final one, 0.25. You can increase it beyond also, but 0.25 maybe we can do and stop. Okay. It's 6.8. Sorry, 6.08. Okay. Now, pausing at this point, is everyone able to follow up till here? What are the experiments we have conducted? Instead of actually running the entire STN GPS system, we thought of it as a random noise generator, increased the variance across the system, and actually looked at how the performance varies. Any quick observations? What happens as the variance increases?
Ma'am, better results, ma'am. It's starting to choose 3 and 4.
Yeah, right? So basically, I'm not saying alone exploration is important. As I already mentioned, value impairment is also a problem with IGT. But right now, we are only looking at variance levels. We are not really impairing the value function, assuming that part of the system is intact. We can think that as the exploration is very, very low, or if the variance is very, very low, the performance is worse. As the variance increases at some optimal level of variance, we will start seeing that the scores turn to be positive. Is
Ma'am,
this observation fine? Yeah,
I have one silly question.
yeah.
So, if you increase the variance beyond a certain point, what happens, ma'am?
It'll be very random. You keep variance as 1, it should be very, very random.
Okay. Yeah,
Yeah.
okay.
So there'll be an optimal level of variance always it should have. It can't be extremely random also. Then it will never exploit it. We'll only keep exploring all the time, which is very weird. So
Is it
there'll be an
possible to
optimal
achieve
level.
here? Sorry. Is it possible to
Uh,
achieve in this one,
I
ma'am?
guess, yeah, if you actually open that IGT code, right? The IGT noise simulator code, you can see how the picks happen in that code. Okay, you keep the variance as 1. You can see which card got picked when. You will see that all 4 cards are being very randomly picked till the end if you keep variance as 1 or 2.
Hmm.
Like here in the Gradio file, I think we are not showing that individually how is the picking happening. You can actually see it in that code. If you want to
Okay.
run it, you can run it and test it also. But then IGT's code doesn't make any sense. It will be turning very much close to 0
Okay.
because it keeps randomly picking all of them equally, right? So
Hmm.
it'll be tending to 0. Yeah. So when I say higher variance, I mean optimal variance. Beyond certain point, it's not good. That's how even any time, right? Exploration should be optimal in nature. You can't keep exploring till the end of the task. I'll only explore in the beginning to some extent. Then I'll start understanding which are the good ones. Any
Yes,
other
ma'am.
queries? Okay. Thank you, Abdul. Thank you so much. You can stop the screen sharing then. Again, maybe we might interrupt you for doing the STN-GPE simulations, but
Okay.
thank you so much. Yeah. Okay. So with this, maybe our first part of understanding the entire IGT system is like clear a little bit. So maybe with that, let me stop before break at this point that wait, one second. Yeah, so what we have observed is as we keep increasing the noise or as there is an optimal performance of the variance, the performance improves. As the variance increases, the performance improves. Okay, so for optimal performance and optimal variance slash exploration is required. Okay. So this is kind of the simulation ka final pointer that we get that at the end of the day, yes, as we keep increasing the variance, the IGT scores improve. And the conclusion is at the optimal level of variance, the system will acquire its optimal performance. Okay. Yeah. Okay, then maybe before we go through the entire STN-GPE system simulation itself, we will take a short break for 10 minutes and then we will resume from there. Is that okay? It's 11:08:09 and now we'll take a break till 11:20 and then we'll start doing the STN-GPE simulations.
Yes, ma'am.
Okay. So guys, please take a break for 10 minutes. Then we'll resume back. After the resuming back session, we will start with the STN-GPE network model, which we are going to model it, do the simulations under PD, calibrate the model and see what happens. Okay, good. Take a break and come back. And the goals of the session. So since we are halfway through, maybe I'll show you this slide now. So the entire thing was kept up in this way that initially you were able to understand the decision-making deficits in PD and how does DBS influence decision-making in PD. When I say influence, I meant experimentally what are the facts that we know. And then we also have the understanding of how does Wessel Ganglia architecture looks like and what is the reinforcement learning perspective of it. And then what is this importance of complexity of STN-GPE dynamics? For now, we are assuming the complexity is variance, but there are more measures for complexity, which we will actually see when we construct the STN-GPE network itself. And then we also did some experiments assuming that STN-GPE is a random number generator or a random noise generator, and then conduct simulations. So going ahead, what are we going to do? We are going to construct more realistic network of STN-GPE system. And then we will calibrate the model to show normal and PD characteristics. Then I will talk about different metrics that we use to classify these characteristics. When I say classify, I mean, how do I define what is complexity? What are the metrics that people use to classify this complexity? Then we understand those metrics basically, and then we'll apply DBS and also check the STM network. So these are the upcoming goals of the session from maybe here. And then at the end, if there is some time permitted, then we will integrate the STN-GPE spiking network into IGT and then run the network as well. We get very similar results, but still, yeah. Maybe I'll move to the correct slide now. Yes. So going to the model of STN-GPE system, as I mentioned, what happens under PD for STN-GPE dynamics? Let's assume this is a 2D grid of neurons of GPE. Generally, we take them to be 16 cross 16 neurons, that is 256 neurons. There is an STN 2D sheet of neurons. GPE connects to STN neurons by inhibitory connections. STN connects to GPE via excitatory connections. GPE also receives input from striatum, if you remember. If you remember the architecture here, the input from striatum goes into GPE as well. Those inputs also we will consider. Yeah, so this is how it looks like. Now, experimentally, what is observed under Parkinsonian condition? So when there is a lower and lower dopamine, as I already said, STN becomes overactive and synchronized. When does that actually happen? As I said that the indirect pathway gets overactivated, that is, D2 striatum will get more and more active. That is, it starts inhibiting GPE more. Basically, your D2 striatal currents are increased. That is, when the striatal currents are very much increased, we will see that PD symptoms show up. And secondly, apart from that, dopamine also influences the lateral connections at STN. When I say lateral connections, apart from STN projecting to GPE neurons, STN neurons also connect among themselves. Like if I take this STN neuron, it connects to some other STN neurons with certain probability. Say out of all the neurons, it connects to some 50% of the neurons. That is what is lateral connectivity. And it connects with certain weight, which I call it as lateral strength. So we'll be looking at these two parameters. One, what happens when I keep increasing the striatal currents? And the other is when I keep increasing the lateral strength, that is the lateral connectivity weight, then it should also show PD condition. When I say PD condition, again, it's that synchronized beta bursting around 15 to 35 hertz. These are the experimental observations. Now, understanding the experimental observations, what do you think my model should be able to do? What should be my model requirements? Can I say the model should be capable of showing both normal and PD condition? That's the first bare minimum of the model, right? It should be capable of showing both normal continuous firing, where you see normal condition, there is a continuous firing, and under PD condition, there is a synchronized bursting. It should be able to show that. And that bursting should be specific to beta synchrony. And when I keep increasing my striatal current, can I say it should be turning more and more as a PD model? And similarly, when I keep increasing the lateral strength, it should be turning as a more and more PD model. Can I say those things? I mean, these are my model requirements. This is how it should behave. Can I say these points? Yes.
Can you repeat, ma'am?
Yeah. So what I meant was experimental observations basically showed that as I increase the external D2 striatum current, it turns more and more PD. Apart from that, if I increase the lateral strength, it will turn more and more PD. Can I say the model which I built should also show such kind of behavior?
Yeah, if you make changes to the like the striate current or the lateral strengths, maybe you can induce PD there.
Yes. So whichever model I will make should have these characteristics. So that is how I will be calibrating or testing my model that the model is correct. Because whenever we do a model, we have to also prove that it is correct. So these are the experimental facts. So my model has to satisfy those experimental parts. Now, did you guys go through any choice of neuron model which is able to show both bursting and continuous firing? If you guys don't understand bursting or continuous firing, let me know. Maybe I'll just brief it out for you guys. Izikevich. Yeah. Okay. So basically, maybe I'll show some result page and then go through it. Just a minute. Yeah. Can you see this one? The first one, the blue plot, it's called continuous firing. And can you see the other plot? It shows that clusters of firing, right? That is what is called as bursting.
Okay.
You guys might have seen the day one Izikevich model, which was capable of actually showing such kind of a behavior. So the choice of neuron model we actually pick is Izikevich. Okay. And even when you actually look at the experimental, maybe I skipped the slide, but I'll let you guys know. If you actually go into the STN and put an electrode into the STN and record a single neuron activity, it looks like this. Can I say this is kind of bursting? Because it fires together, stops for a while, fires together, stops for a while, fires together, stops for a while. It's noisy because I have been recording it using an external electrode, but this is how a single neuron activity looks like. Now, apart from single neuron activity, what we do is when I keep an electrode, it might record some cluster of neuron activity, like all the net cluster voltage together. That is what we call it as local field potential. And this is how it looks like. If I actually do a frequency analysis of it, this is what the frequency looks like. It shows peaks at 15 and around 25 hertz. This is what we meant to be that it has some beta frequency within itself. Does it make sense?
Yes, ma'am.
This is an experimental data which I picked from Little and Brown 2016. Okay. LFP is a cluster of burst. Yes, Abdul. Basically, what happens is if I have 100 neurons together and put an electrode in it, all the neurons' activity gets captured within that. So we call it as local field potential instead of measuring from a single neuron. Yeah, cool. So just entering to the model of the STN GPE system. So this is how we model the entire STN GPE system. I take a 16 cross 16 grid of STN and GPE neurons. And each of the neuron is modeled as an Izikevich neuron. You remember that two-dimensional Izikevich equation, right? I model both of them as Izikevich neuron. GPE gets currents from striatum and STN and also its lateral currents, whereas STN gets currents from its lateral currents as well as GPE. Okay, and all the connections are made to be random. That is, if I take one neuron here, it will be randomly connected to 70% of the other neurons. Same thing with STN here. It will be randomly connected to 70% of the other neurons with certain weight, lateral weight. Is that okay? If you want to actually look at the detailed equations of this STN GPE network, it's actually a very huge biophysiological model. You can check it out in the paper we have sent you guys. In the paper, it gives you the full equation. So each neuron is modeled using this Izikevich equation, where it actually receives the external currents. And these external currents are your receptor currents. STN has these receptors, NMDA receptors and AMPA receptors. GPE has GABA receptors. So they model it in a very, very detailed fashion. But maybe you guys can have a skim through it. Not necessarily you can understand each and every equation of it. That's okay for today's session. But you can just imagine that each of them has receptors. All the receptors have some equations that result to some current that gets integrated into your Izikevich equation. That is what we do. In the same code repository, the code can be found here. One second. Yeah, the code can be found within the STN-GPE folder. It has this stngpe.py file, which actually has that STN-GPE loop simulation. It also does that very simple Euler equation solving. For every dt, it solves and just appends all the values. Is that okay? I'm not going through each and every line of the code because it's a very standard Izikevich code along with other receptor equations. That's it. Okay. Cool. Then maybe I'll go into this slide. Yeah. So this is how we end up modeling your STN GPE system. Now, how do I model deep brain stimulation to it? So as I mentioned, deep brain stimulation is a surgical surgical thing where they actually put electrode into the STN and start stimulating it with electric pulses. And these electric pulses are basically biphasic in nature. Any idea what is biphasic? Basically, for every positive pulse, we also give a negative pulse so that there is no charge imbalance that gets created. If there's a charge accumulation that gets created, it will create cell death. So we try to not maintain charge accumulation. So if I have to model the STN DBS model, I will take the same STN grid in that previous model and keep an electrode at the center. or any place you want to. For us, we are assuming it is at the center, and this gives out these currents, which are these pulsatile current with a frequency of 130 hertz, amplitude of 220 picoampere. And to characterize any pulse, I have to define the pulse to pulse interval, that is this delta. What is the difference? And also duty cycle or a pulse width. That will give me all the parameters for the biphasic stimulation, and I stimulate it. Now, the question is, if I keep an electrode at the center, do you think the current effect at the center versus to the end of the sheet will be the same or different? What do you think? What I mean is, I'm keeping an electrode here, then the current that I stimulate at this neuron versus the effect of that current at this neuron, will it be the same or different? Yes, center will have a higher intensity in comparison with the outer. That is, if I'm stimulating here, with the distance, the intensity of the current decreases. Do you have any mathematical formulation which shows such kind of a behavior that at center it's high? As you move across the distance, it will get reduced. Any mathematical way of modeling that spread of current? Any guesses? Doppler, yeah, but what I mean is in terms of if I want to just model a spread equation in 2 dimensions, can I say I can put a Gaussian? A simple Gaussian can make sure that it is highest at the center and lesser towards the ends, right? So we tend to model the spread as a Gaussian spread. That is, at the center it will be high. As the distance increases, the spread effect will get reduced. Is that okay? Maybe. With that, then we will go into the code simulation of ST&GPE system. So the ST&GPE system takes quite some time to simulate, at least 2 minutes per simulation. And we are having some 5 to 6 simulations to be done. So we will be doing it slowly. There's no hurry to it. We'll try to do each simulation slowly and understand the metrics. First, I will do one simulation and show you guys how the metrics look like. And then we will have someone to share the screen and complete the full task. Okay. Let me go to the code file. So here also to run the code simulations, there are 2 code files present. One is run STNGP. It's a blank code where you can change the parameters and run. There is also an interface version of it, which I will be using for today's simulation. You can open the interface simulation itself. In the interface simulation, again, it clones onto the same repository so that it can inherit the functions from this. And then it opens up a simulator for you guys. Are you guys able to open it? If you guys are facing any issues, let us know. It's just that STNGP radio file, you just run it from top to bottom. It will create a simulator link. Click open that simulator link. From there, I'll show you guys what to do. Is everyone able to open the simulator page? Thanks, Abdul. I'll wait for a minute so that everyone is able to access it. Once it opens, it looks like this. Okay, I'm assuming everyone is able to open it. So once you actually open it, it tends to look like this. This is the kind of simulator page you will have. The left side of the parameters are for the entire network parameters. The right side is the DBS parameters. So if you checkbox the DBS, it will run it with DBS. If you don't checkbox it, it will run it with the normal parameters. So right now, I'm not checkboxing it for now. Okay. So what are the parameters that we define here? One, how many STN GPE units we need. I am fixing it at 16 because the model I have calibrated is for 16 cross 16 neurons. If you increase it, you might have to recalibrate the entire system. So don't do that for now. This time steps and dt are for solving your differential equations using Euler. And lateral sparsity and intersparsity will define how many neurons it will connect. That is, out of 100 neurons, it will be like connecting to some 90% of the neurons. Then I have these striatal currents. You remember, I'll be having an input current into the GPE from striatum. That is this, I STR D2 GPE. I'll also have a lateral strength of STN parameter. I'll also have a lateral strength of GPE parameter. WSG, what is the strength that STN connects to GPE? What is the strength that GPE connects to STN? And some external parameters for analysis. Are the parameters clear here? I mean, though we are not going to play around with all the parameters here, it will be a mess if we change all the parameters all together at once. We'll only be changing these two parameters. That is STR D2 GPE and lateral strength STN. These are the only two parameters we will keep changing. Is that okay? And below that, there is a run simulation button. If you run it, it will take a minute or two to do a simulation, and it shows up plots like this. And print some metrics here. Okay. For today, let's only focus on STN. Let's not deal with GPE. The STN plots here are on the left side. I'll go through each and every plot if everyone is ready for it. Can I go through the plots? I'll just explain the plots, then we will do all the simulations together and understand what is going on. Okay, let me start with explaining the plot. The first plot here is a single neuron voltage. This is a continuous firing. When you change parameters, you will also see bursting condition later, but this is kind of a continuous firing. The second one is LFP. So what we do is in the 2D grid of neurons, we will take that LFP potential and calculate it and plot it. The third one is raster scan. Do you guys understand what is meant by a raster scan in general? Or have come across somewhere in your previous time?
No.
Okay, so raster scan is basically kind of shows how neurons are active at each time point. Right now, I'm not able to zoom it in, but here the x-axis is basically time and the y-axis is a neuron. So this is 0th neuron, 1st neuron, 6th neuron, 10th neuron, 100th neuron, 200th neuron. Like that, we have 256 neurons. If I go like this horizontally, it will tell me at 256th neuron, when was it active? That is, it is firing at each of these time dots. Each dot represents it fired at that point. So it fired here, it fired here, it fired here. Does that make sense? All of you?
Yes, ma'am.
Okay. Across the time also, you can see if I go across this time, I can pinpoint, okay, this neuron fired at this point, this neuron fired at this point, this neuron fired at this point. Okay, you'll see a distinction in raster scan when you actually see it with PD condition. That will make more sense at that time. Why do we look at raster scans? But yeah, right now you can assume this is pretty much randomly firing. You can't pinpoint if there is any trend to firing, at least here. You'll see a very patterned trend when you see the raster scan of PD. The next one is basically spectrogram. The x-axis is time. Across time, which frequencies are active? You're not really seeing any frequency band active here. If you look at in PD condition, it will show a very strong frequency band active. Maybe I'll also show how PD looks like for you guys to understand it better. This is how PD looks like. Let me present my screen. Okay, so this is how PD looks like. Under PD condition, you will see these bursting. And can you see the LFP activity is also different now? In the previous one, the LFP activity was very much like randomly seen. But whereas here it's very periodic, very synchronized. Even under raster scan, if you look at it, you see that there are these neurons which are firing together in clusters. I can only see cluster firing here. Can you guys observe that? I'm sure that is very easy to observe. So this is what I call it as synchrony. All the neurons are here firing together and stopping together. Here in the previous condition, here they are randomly firing, whereas here it's very much synchronized. And if you look at the FFT spectrum or the spectrogram itself, it shows strong band at these two frequencies, which are basically beta frequency. So if you keep changing the parameters, these kinds of effects actually come up. You can see these kinds of effects. Apart from these, in the same code file, if you actually go through it a little below, it also prints metrics. One is entropy, synchrony, and standard deviation. Standard deviation is that if I have full neurons, I'll take 4 clusters of neurons activity and see what is the standard deviation or variance across those 4 activities. That is standard deviation, which we have also used before. The other 2 metrics are synchrony and entropy. These are the 3 metrics which we are using to calibrate your complexity. Standard deviation we already know. The other 2 metrics are entropy and synchrony. Do you guys understand by the term what is an entropy in general?
Yes, ma'am.
Yeah, it is kind of a measure of randomness, right? Now, there's something called as a spectral entropy. What is a spectral entropy? Maybe I'll give you an intuitive understanding. Mathematical formula you can actually understand later. So as an intuitive understanding, if you want to look at what is a spectral entropy, if I take a FFT spectrum, if there is a very, very sharp FFT spectrum, that is sharp peak at one frequency, then it is low entropy. It is very periodic. It is having very less entropy. Whereas if the frequency spectrum is broad enough, then we call it as high entropy. Now, as I already mentioned, PD people show sharp frequency peaks at beta frequency. So their entropy is actually lesser in general. And under normal condition, we will be seeing a higher entropy. This is the intuition behind what is entropy. Similarly, for synchrony, I think I have already explained before also. Synchrony will tell me how synchronized each neuron is firing. That is, if you actually look at this, you can see two neurons fire together anytime. They're very random. So I call it as low synchrony. Whereas in this condition, which is like bursting or PD condition, we can see all the neurons firing together, which is high synchrony. So we'll be seeing normal condition low synchrony and PD condition as high synchrony in general. Is that clear? Are you guys able to understand what are the metrics we have seen at least? So these are the 4 to 5 things which we use to calibrate our network. Or these are the 3 things which we use or term it as complexity. Someone was asking what was complexity, right? So this is actually what we term it as complexity, all of 3 ones. Is it clear?
Ma'am?
Yeah.
So if I get it right, so at a high spectral entropy, the frequency may be chaotic, and at low, it will be like very orderly. Is it?
Yes, you're right.
Okay.
If you have gone through the oscillators and chaotic systems, yeah, you're right. That is how it looks. Or in general, you can think of the STN dynamics under normal condition to be more chaotic and under PD condition to be more synchronized.
Okay, ma'am. Thank you.
Yeah. So with this background, can we go through some simulation sessions? By simulation, what I mean is, remember before I said that we need to calibrate the model? When I said calibrating the model, I mentioned that as I increase the striatal currents, I have to see a transition from normal to PD. Similarly, as I keep increasing the lateral strength, I should see a transition from normal to PD. And then we can do the DBS experiments. So these are the coding simulations which we are actually going to do. Yeah, so these are the tasks that are to be done. Is anyone willing to share your screen? I'll guide through what are the simulations and how can we do it. Maybe Abdul again, I don't know. Is it okay for you, Abdul? Because you have the PPT also with you to make a note of all the values.
Yeah, it's fine, ma'am.
Yeah. Can you just share the screen then?
Just a minute.
Yeah, sure. It'll take a while to do all these STNGP simulations, so just be a little patient. I think each simulation takes around 2 minutes. So around 10 to 15 minutes it will take for us to do the simulation. Yeah. Can you also go into the PPT? Yeah, just go down a little. Code simulations, a little more down. Yeah, so I have put up the tasks as 1A, B, and 2. Under first 1A task, we will increase the striatal current and observe the transition. Under 1B, we will see increase the lateral strength and see the observation. And then we will do the DBS measures. Okay. Maybe you can go to the next slide. Yeah. So for the first one, what we will do is we will fix the lateral strength, increase the striatal current, and see what happens to the metrics. You can go to the Gradio page. I can dictate the parameters to you. You can actually load it up. Yeah, this one, right? Yeah. You can keep the lateral strength of STN to be 0.0275. Yeah. Now, we will fix the lateral strength for this and the next simulation at this value and increase the current. First, we will run the simulation for a current of 0.5. Then we will increase it further. Okay. Yeah. You can run the simulation.
Ma'am, can I close the other one which you did before?
Yeah,
Would it
you
be?
can. Yeah, the other Gradio IGT one, right? Yeah, sure.
Mm.
We might need it later, but then we will figure out later. Right now, we might not need it. The thing is, if you run it on your local system and it has a higher RAM, it runs pretty much faster. But when you do it on Colab, it takes a little time. It depends on your internet speed in general.
Oh.
So that's okay. I mean, this is the time it takes for me on Colab as well. It runs how many? Almost 256 into 8 differential equations every time. And it runs it for some 50,000 times. So it takes quite some time.
Okay.
Yeah.
Uh, I don't know if this is a random question, but is there a
Yeah.
specific reason you use Mac over a Windows system? Is that a
In
RAM
this,
limitation?
I think so. I don't know. Right now, when I used to use Windows, it was pretty much slower. With Mac, it was faster because of its dynamic memory system. But I don't know.
Okay.
Again, it depends. Yeah. So can you go down? For the lower striatal current, that is 0.5, what is the kind of behavior are you thinking is this? Is this a normal behavior or a PD behavior?
Normal.
It's a normal behavior, right? And if you look at the metrics, can you observe that STN is showing higher entropy? That is, it's more random in nature, very low synchrony, and higher standard deviation. Yes?
Yes, ma'am.
Okay. Then we'll do one thing. We'll load these numbers into your PPT so that we can get back to it and have a reference over it later. Can you open the PPT? I noted down the parameters. I can tell you. There is a PPT table that is present on page 16. Yeah. So for the lateral strength 0.025, and when I ran it with a striatal current of 0.5, we observed continuous firing. Maybe you can write continuous and bracket, you can put it as normal. Yeah. Entropy we observed is, one second, what happened? 0.82, sorry, 0.884. Yeah, synchrony, maybe I'll 0.084. And standard deviation is 0.28. Yeah. Now, after this, we will increase the striatal current to 5, right? Can you run the simulation? Yeah, it takes quite some time. And meanwhile, if anyone is having any more queries, you can ask us right now. How the performance wise, different code, different running a code in one. Arup, it should be very similar. If you run the normal code also, it does the same thing. I mean, on simulator, it's just the front end cap I have put. Back end, it runs this very same code. Maybe you look at the correct parameters you have to change and run. And make sure your DBS is false. It should give very similar results. Yeah. So what does the performance look like now?
It has become synchronous, man.
Yeah, it has become very much synchronous, which we can actually observe it when we are looking at it in the raster scan also, right?
Yeah.
And also it gives very strong beta peaks. Even LFP looks very, very periodic in a way. It's a noisy periodic, but definitely periodic. Yeah, maybe then we will load up the parameters in your values in the PPT. I can dictate it. I click the picture of it. So it's bursting and you can write PD there. Yes. Entropy is 0.3. Synchrony is 0.69. And standard deviation is 0.07. Yes. So quick observations. Can you see that under PD, the entropy is decreased and synchrony is increased? Variance is also decreased. So we can see that for a given lateral strength, as I keep increasing the external current, it moves from normal to PD. Okay. We can go to the next simulation then. The next part of the task is to keep the striatal currents constant, change my lateral strength. I'll go from lateral strength of 0.02 to 0.03. Yeah. Keep the lateral strength as 5. Lateral strength as 0.02 and external current as 5. Yeah, you can run it here. Okay. Yeah, it finished running, right? What is the behavior do we observe now? It's continuous firing. Basically a
Normal.
normal kind of a behavior, right? Yeah. We can maybe then save the parameters for now. We'll go back to the parameters when we are doing the comparison analysis. Yeah, continuous, normal. Entropy is 0.87. Synchrony is 0.118. And standard deviation 0.23. Yes. Now we have to increase the lateral strength to 0.03 and run it. Yeah. Also, meanwhile, can you share that presentation with me? Maybe I can use those numbers to like discuss the pointers. Maybe anyone with the link can be able to view it.
Yeah, this link, ma'am.
Yeah, you can go to the share and do it. Otherwise, I don't think I'll be able to view it. Yeah, share. Yeah, anyone with the link. Yeah. Can you put it in the chat box? I'll
Yeah.
take it from there. Good. Thank you so much. Yeah, I think I'm able to access it.
Ma'am,
Yes.
so what happens when you do deep brain stimulation in the STN, ma'am?
You will see it now. The last experiment will be doing DBS. It will actually try to reduce your synchrony, improve the entropy and also standard deviation, but it can't do it to a lot of extent. It only do it very minimally. I mean, it doesn't achieve till the normal condition, which is what we are expecting why DBS doesn't work for decision making. You'll see that in a while. Yeah. Yeah. What is the kind of behavior we see now? We end up seeing PD behavior, right? That is, as I keep increasing my lateral strength, it turns out to be, it is bursting very synchronized. Yeah, you can go back. We can put up the metrics. Yeah. Entropy is 0.26. Synchrony is 0.674. And standard deviation is 0.06. Yeah. Okay. Then the next one is to run it with DBS. But for the same parameters, we will run it and see instead of trying out others. You can go to the same Gradio page and click tick mark the DBS. Right side. Yeah. Under DBS function, there are different functions, but right now only biphasic DBS works. So don't play around with the other ones. Then you can define DBS frequency, duty, amplitude, pulse interval. And the last three parameters, DBS center tells me where to keep the center, where the electrode is placed. What is the spread amplitude and what is the spread sigma? It's basically to define that current spread. We'll keep it at these numbers and maybe run it for now. Yeah, you can simulate it. You can run the simulation. Yeah.
Ma'am, you said that during
Mhm.
DBS, the current pulses would be given to the STM, right?
Mhm. Yeah.
Would it be like based on an impulse or like on a random basis or like periodic basis?
So the pulse looks like that periodic pulse which I've shown in the figure, right? That biphasic pulse. Can you just Google such biphasic pulses? Pulses. Yeah, it looks like that.
Okay. No,
Yeah,
I'm asking for the patient, would it be like a constant stimulation or like it would be periodic?
it'll be periodic.
Okay.
It's not a constant current. It'll be a periodic, some 130 hertz is what clinically they do. They'll also
Mhm.
slightly reduce and increase the frequency. Right now, people are trying to see if low frequencies can work because it's better to do low frequency in terms of cells. Performance hasn't shown much improvement with low frequency, but in terms of cell oxidation and cell death, that is better.
Okay.
Yeah. Yeah, maybe wait, let me just capture the metrics first. Yeah. So can you see that the bursting is slightly disrupted here in a way? I'm not saying fully disrupted. If you look at the raster scan, it's more spread out towards the center, right? It's trying to disrupt it there. And also the high end frequency that is around some 25 to 30 hertz, which was a very more thicker band, now has slightly vanished away after giving DBS. Though that 15 hertz still retains, but at least that 25 hertz is slightly removed right now. Can you guys make that observation?
Yes, ma'am.
Yeah. Maybe
Kind of.
then we'll write, yeah, it's not very great, but it does to some extent. If you actually go and load the metrics, maybe then you can compare it better. I'll dictate the metrics. Yeah. Just give it as PD DBS. It's not really bursting, not even continuous altogether. Okay. Entropy is 0.329. Synchrony is 0.54. So 0.45. And 0.08 is the variance. Okay. So now from PD bursting, where the entropy was 0.26, PD DBS increased the entropy. Yes. With respect to PD, yes, it improved the entropy. Similarly, it also reduced the synchrony from 0.674 to 0.45. I'm just looking at the last two rows. Yes,
Yes, ma'am.
maybe, yeah. And also with respect to the variance, can you see that it increased the variance? It did what DBS has to do, but is it as good as a normal condition? Compare it with any normal condition and see. Is it as good as with normal condition?
No, slightly
Not
better
really.
maybe.
Yeah. So generally, motor symptoms are able to improve because it reduced the synchrony. It was able to reduce that 20 hertz peak because of which the motor symptoms really did improve. But for decision making, it requires that normal level of variance in general to be improved, which I guess DBS doesn't really do it. Which is a hypothesis we think that why DBS doesn't really work with Parkinson's patients. I mean, along with this, there is also value impairment, which we are not touching, but still, this is what is the approximate hypothesis. Does
What do you
that
mean
make
by
a
that?
little,
Sorry.
yeah?
What do you mean by value impairment? In terms of
So you remember when we were training the network, there was this TD error which we used to back propagate, right? For learning the value.
yeah.
So that is also dopamine. That also gets limited. But the only thing is the limitation that happens to the synchrony and that value impairment is fractional. When cell death happens, both happen at different levels. So we are right now imagining that the values are still intact and only exploration is lost. If value is lost, DBS can't restore it. That's a given fact.
So it's
If
like an
value
early
is completely lost.
stage of
Yeah.
disease.
Yeah, in the early stage, if you do DBS, there is a chance that it might improve, but not even with the standard DBS. If the standard DBS is slightly modified such that my variance, synchrony, and all go back to normal condition, then you can improve it.
Okay, ma'am.
Yeah.
Ma'am,
Ah.
so let's say instead of DBS, or
Mhm.
is there anything that they give L-Dopa and all that's to improve the dopamine levels in the system, no, ma'am?
Yeah, so L-Dopa also has a problem. The thing is, when I give L-Dopa, I don't know what is the level of cell death I have already undergone. So there might be an excess dopamine that gets into the system because of L-Dopa. So they will have opposite effects. They'll start becoming extremely impulsive and extreme rapid movements they'll start making. And it also has this wear off effect. That is, after a while, L-Dopa doesn't start working well. That is when they actually do deep brain stimulation. So deep brain stimulation is only done in the later phases of the PD. So it's going to be very difficult to improve decision making if you're doing it in the later phases of the PD. You can only improve it given it can restore the variance and given the values are still intact.
Okay, ma'am. Okay.
Yeah. So this is actually a kind of a problem. We can take it forward from this, that how do you use a deep brain stimulation? What kind of stimulation is good to improve such kind of a variance? That can be one of the problem statements you can build out of it. Because since the current stimulation is not very great. When I say current stimulation is not very great, can you go back to the variance levels you saw under under your IGT task? How did the variance levels show the value? Yeah. Can you see when the variance is around 0.07 or 0.05, the scores were negative, whereas around 0.2, the score is positive. Can you correlate those numbers with your PD DBS as well as normal condition now? Even without running the entire spiking code, can we estimate that this is how probably the entire system IGT scores will look like? Does it make sense? I'm just saying that STN produced a variance of around 0.05 under PD. So approximately my score will be negative, right? If I even run the entire IGT code, it's going to be negative. Similarly, under normal condition, my variance was around 0.25. So it is expected that the values will be somewhere like positive. If you want to run it with spiking network, maybe we will do it for one run. Should we try it by integrating the spiking network and doing it without noise?
Yes, ma'am.
Can you go to that IGT Gradio page? It will take 5 minutes at least to run the simulations. That IGT Gradio code.
Should I open that again?
I think you opened it already. It's on run. You can open that only. The next one.
This
No, next one. Next one. Yeah. Go down. Can you just go up and open that host page? Yeah, open it. Yeah. Now what we will do is we will run it with STNGP. You can tick mark that STNGP under normal condition. Let's just run it. When we are clicking STNGP, the variance has no thing. First, it will actually run that STNGP network for some 50,000 steps. Then only it does IGT. So it might take a little while. Just wait for it. And also we have to do these runs at least like 4 to 5 times under normal condition. Take an average across it to actually see a proper trend across it. Because, I mean, since the variance will be there, there will be different kinds of performance. But overall, when you do it like 4 to 5 times, you will see that there is a positive score at the end. Yeah. Can you see that the IGT scores are now positive when you actually run it with the normal condition?
Smell.
Yeah. You can similarly do for PD. Maybe we'll stop with PD and PD DBS if time permits, and then we'll conclude the session. We can run it for PD. It's taking like 2 to 3 minutes. Okay. I'm also running it in parallel. Yeah. Can you see that the values are negative now when you run it with PD condition? That is because its variance is not high enough that it can actually improve your IGT scores. Similarly, we can do it for PD DBS as well. You want to try it now?
Yes, ma'am.
Yeah. Standard DBS, yeah. It might slightly be better than PD, but there's no necessity that it will improve very much as normal condition.
Uh, is there any other type of cure for PD? As far as I've seen, there's no cure or something. Like, is there anything like that, ma'am?
Sorry?
Is there a
Cure
cure for
for
PD? PDM.
cure as in like completely revert back to symptoms?
Maybe like
Mm,
similar to normal, perhaps.
That might be a little difficult because medication also, then you have to understand what is the level of dopamine that is depleted and give
Mhm.
only that level of dopamine so it doesn't get overactivated or over usage of medicine.
Oh.
And DBS also has its own side effects. We can only control symptoms to some extent, but we can't really cure it unless and until some kind of gene therapy works and you can revert back the cell death. But that is not really possible. But there is one hypothesis that maybe doing deep brain stimulation at the initial phases of PD might
Mhm.
reduce cell death. But that's
Oh,
still under hypothesis. I don't know how much of testing has been done on that.
okay.
Yeah. Yeah. Can you see that the scores are lower now? It's just 1. It doesn't even reach 5 to 6. If you rerun it, it will also show negative scores if you run it again also. I mean, if you want, you can rerun it and check it. That's because of the variance it has. But again, the scores are not as good as normal again. Does that make sense? Sorry?
No, I understand, ma'am.
Yeah. Okay.
So if we change anything related to like the DBS amount, would we get a better result?
So if it's a very mild PD condition,
Hmm.
then you can, like, if my lateral strength is at the border where it is just transitioning from normal to PD, then it does give you better results.
But PD as such is a progressive condition, isn't it? Slowly the neurons
Yeah.
will die.
Yeah. So if you can identify PD in very early stages, then yes. As I mentioned, the PD symptoms would only show up when your almost 70% cells are dead. So that's very tricky. If you can identify it in very early stages, maybe yes, we can do something about it. But in later phases, I don't think you can do anything about it. Okay. Maybe then I'll start concluding the points, whichever we have observed till here. Thank
Can
you,
I
Abdul.
stop
Maybe
sharing?
I'll share. Yeah, maybe I'll share the screen and conclude the points here. Yeah. Is my screen visible now?
Yes, ma'am.
Okay. Cool. So to conclude up the points, in this specific task, we have ran the system under normal PD conditions as well as PD DBS, and we were able to calibrate the system. That is, as I keep increasing my striatal currents, it moves from normal to PD. Similarly, as I keep increasing my lateral strength, it moves from normal to PD. Whereas under PD DBS condition, the variance is not very, very improved. And the synchrony and entropy also improves with respect to PD, but not as good as normal condition. Generally, when experiments talk about it, they always talk about it with respect to PD condition. They say with respect to PD, DBS is improved, but did it improve as much as normal? Not really. And that is one thing we have observed over here. Okay, so maybe let me go back to my presentation and put up the stuff. Yeah, so cool. To further conclude the points, that is what we have observed. And can we correlate the variances we observed with the noise experiments we have done with IGT? Yes, we have done that, right? And looking at that, we were able to guess that normal will definitely have a better performance in comparison with PD or PD DBS. And we have also done some simulations with your by integrating it with spiking network. And when we did that, we didn't really see as good as improvement to normal condition. And finally, if we come to the conclusion of the session and to just summarize if we finished all the goals or not, yes, we have started with understanding the decision making deficits, BG architecture and RL perspective. And then we also looked at what is the complexity of STN-GPE dynamics and how it contributes to decision making. And then we said, okay, fine, let me replace the entire STN-GPE system as a random noise generator and conduct the experiments. After that, we have actually built up, instead of a random noise generator, we have built up a complete spiking network of STN-GPE to show PD and normal characteristics, and also conducted DBS experiments on it. And then we have also conducted the variance level experiments and replaced it with spiking network and observed the IGT results. So if you want to think about any further perspective of how can we go ahead with this system and all, yes, the go ahead with this thing would be to identify what kind of protocols can improve the performance as good as normal, or how can we improve the performance or the metrics or the complexity as good as normal. We can try out different kinds of stimulations. People are trying out some kind of closed loop DBS or some RL controller loop DBS and stuff, which might still work better. So yeah, I think that brings us to kind of an end of the session. If you guys have any pointers to discuss, you can let us know.
Is there anything else that is done in synchrony with DBS, something like TMS or something
Uh,
that would help?
TMS, so the one difficulty with TMS is it is given at the scalp level. And basal ganglia and STN are located very deep inside the cortex. So TMS is not very effective for this thing. People were trying to see if ultrasound can be used for it. I don't think they have hit any luck with that either. The only problem with this is that it's very deeply located. It's like very deep inside the cortex. So even measuring the STN signals from cortex, like in EEG and all, is very difficult. You can't actually pick up those beta synchrony and all at EEG level very easily. People are trying to see how to do it, but I don't think there's any luck yet on those lines.
Okay.
Yeah. Okay.
Hello.
Yes.
Yeah, so first of all, thank you for this wonderful session. It was really interacting and very helpful. So, just my quick question. I'm very much interested in ultrasound stimulation
Uh-huh.
and understanding decision making through that. So, have this model ever been used to understand like the way deep brain stimulation you were showing? Have this
Yeah.
also been used understanding ultrasound stimulation and how it
Um,
can help?
you mean have we tried out understanding the ultrasound
Yeah,
stimulation at this? Not
yes,
really.
and if there are like other labs that you have come across that are using this.
I have only read up an article some time ago that people are still trying it. Like, I don't think anyone has tried out modeling behavior of it. Because the thing with ultrasound would be to how do you alone isolate STN and alone stimulate STN?
Hmm.
That's a problem. With DBS, the electrode is exactly located in STN, so it will be able to stimulate it. Ultrasound is not an invasive procedure. So, how do you control it? I'm not very sure. So, unless and
Hmm.
until we know how the hardware works, it might not be really possible for us to model it in the model.
Okay, okay.
Yeah.
Yes. Yeah, thank you. Thank
Thank
you so
you.
much.
Thank you. Okay. If there are no more doubts, maybe I can wrap it up here with the project session. You guys can always contact us back if you guys have any queries. You can write emails to us if you want to discuss anything over it. And let us know if you guys have any troubles with the code also in later times.
Hello, ma'am.
Yes, Abdul.
Uh,
I can't hear you.
Hello. Yeah,
Yeah, I can
hope
hear
so
you
now.
now. Yeah. Yeah.
Can I have just 5 minutes? Like, I need
Yeah,
some
sure.
clarification in a breakout
Yeah,
room or
sure.
something. Probably not here
Uh,
since people are there.
yeah, maybe if everyone is done, then we will wrap up the session and then we can stay back and discuss your queries.
Okay.
Yeah. Okay, guys, should we write email to Mukesh? It's maybe not to Neurogati. You can write to any of us. We are also added into the email threads. So you can write to any of us. Or the CNS Lab website itself has our email IDs. You can write to us based on the projects we work on. Or at least to Sir, Professor Srinivas Chakravarti, he will also forward it to us. Probably Neurogati email, we might not be looking at these kinds of things. So, yeah. It does work. Wait, let me see if it is not working. Oh, maybe they'll try to fix it. I'll ask them to fix it up. I think there might be some issue with it because of which they're not able to do it. I'll ask them to fix it up. That won't be a trouble. Then you will be able to. Yeah, cool. Then thank you guys. Thank you so much. If you guys are done, then we will wrap up the session at this point. Abdul, I'll be here for like 5 more minutes. We can have a discussion over whatever you want to
Okay.
discuss. Yeah.
Okay.